So good afternoon, everyone. Just before, again, we start, for anyone new, we asked everyone to turn off their cameras and microphones just to ensure good video and sound quality during the presentation. And then after, you can turn them back on for the question and discussion period. So today we have with us Dr. Yuri Utsunomiya. Dr. Utsunomiya holds DVM, MSc, and PhD degrees in veterinary medicine from the Sao Paulo State University. During his undergraduate studies, he worked for the Bovine Genome Sequencing and Analysis Consortium in the annotation of the first bovine reference genome assembly. During his graduate studies, he worked for the Zebu Genome Consortium, an international initiative that pioneered the implementation of genomic selection in Zebu cattle in Brazil. He was also a visiting scholar in many labs abroad, including in Italy, UK, USA, and Austria. After finishing his PhD, Dr. Utsunomiya became a lecturer for the graduate program in animal science for the School of Veterinary Medicine of Aracatuba, where he teaches statistics and bioinformatics, apart from supervising graduate students. He serves as associate editor for animal genetics, as well as an expert consultant in international missions organized by the joint FAO IAEA program to deliver training in genomic data analysis in research institutes in South America. In the private sector, Dr. Utsunomiya worked as a consultant and a shareholder at Agro Partners Consulting, a startup company based in Brazil that deploys genomics in agribusiness. And in 2022, he joined ST Genetics as a senior genomics research scientist, where he designs, develops, and deploys genomic-derived technologies, products, and services for the dairy and beef cattle industries. So thank you for joining us again, and I'll give the floor over to you.
Thank you very much. So I'll just turn off my camera as well because my connection is not very good today, but I'll come back later after the presentation. So, oops, I clicked the wrong place here. So you can see my screen again back?
Yeah, we can see.
Okay, perfect. Yeah, that's fine. All right, so I'd like to start by saying that's true privilege and honor for me to be here today talking to you guys, because this is a group that I have followed the scientific footsteps for so many years now. And I had the great fortune to have collaborated with, or at least interacted with, some of its members. And it's really a blast to be here today. Thank you very much for this opportunity. So I picked this rather broad and kind of a pretentious sounding title for the presentation without really realizing it, because I had a deadline to deliver a title for the presentation. So it's genomics in animal breeding, transitioning from basic science to applied technology. But this is far from being an overview of the field. I don't have the necessary seniority for that. It's just me trying to say that I'm going to talk about things that sound like basic science, things that are familiar to you guys, but they are actually at the frontier of things that can be applied to the industry and to the field. So hopefully this will sound fresh yet familiar to you guys. I'm going to start by just giving you a brief overview of what is ST Genetics, the company I've been working for since 2022, and brief overview of our genetics and genomics team. So ST is a big company, has presence in many countries nowadays. It's based in Texas, in Navasota, near College Station. And it has a large umbrella of things going on, but basically we can map the initiatives of the company to four main components here. So we have our proprietary gender selection technologies. So basically sex-sorted semen. We do sorting of semen based on molecular weight. So if you guys remember, X chromosome is a very large chromosome. The Y chromosome is a very small one. So we can have a differential of mass between these two populations of sperm cells. So using flow cytometry, we can separate them, and cell seaming that is going to give you a given percentage of female animals being born or male animals being born. We can do either way. So we have this collection of technologies that serve this kind of purpose. Then we have the dairy and beef genetics. So we don't only sell semen and provide the service, but we also own the genetics. And we have programs, our largest program is in dairy, but we also have a very nice presence in beef and beef on dairy nowadays. That's where I kind of live now today. We also have the same kind of a situation in swine. So we have AccuFast for porcine genetics, and all of that come together because we also have a laboratory that has all the portfolio of things, genomics, that is going on nowadays, called Genetic Visions. It's based in Madison, Wisconsin. So going to briefly talk about more, I will talk more about the dairy and beef genetics, but we will also talk a little bit about genetic visions as well. This is the team. I must say that this is a group of very talented, brilliant people. If you guys don't know these guys, you should probably get to know them better and get in contact and share some experience with them. They are very nice and very easygoing people. I must say that the fact that 80% of the team is Brazilian makes a lot of difference in that department. But we're a very diverse team. We have people whose nationality is from China, we have from Nepal, the United States, and so on. And we are led by our technical director of genetics and genomics, Joseph Deeb. He's a very nice person, very open, and he's always pushing us to the limits of what we can offer and do. So that's the group of people I work with, and this is how we see today the portfolio of things that the company has to offer to the market, articulating with the research we do in our team. So at the core of the business, we have the sex sorted semen technology. So I'm just going through this wheel clockwise here, which I just briefly talked about in the last slide. We have a program because we produce our own genetics. We have a program in reproductive biology and assisted reproductive technologies called GIVIT, Juvenile In Vitro Embryo Transfer, which is a technology that allows us to do ovum pickup in heifers as young as 3 months of age. If you guys remember the breeder's equation, genetic change being a function of the reliability of your evaluations times the selection intensity times the genetic variation you have divided by the generation interval. So that's where the impact of this program lies. So we reduce the generation interval tremendously. And so therefore we can do very fast genetic progress within the company. If you think about a 3-month age heifer contributing with oocytes, then we do in vitro fertilization, embryo transfer, 9 months of gestation length. We have a total of 12 months generation interval within the company. That's really crazy. That's the first place I've seen anything like that. So we are very proud of this advancements we're doing in fast-pacing genetic progress. Of course, that this is all coming together because we have genomic predictions for a number of traits. We have not only for dairy, but also beef on dairy, where most of the phenotypes are collected on F1 crosses. So it's a very interesting program we have there. We have genetic visions again that provides us with a lot of data. They have SNP arrays, next generation sequencing. We can do short reads, long reads, genotype by sequencing. Everything's possible there. And of course, we are living in the moment of deep phenotyping and sensor data. We have a whole division within ST for that, the FarmFit. Last year, we just launched our bolus device, which can capture in real time rumen temperature and rumen movement and track animal health and performance. And within the deep phenotyping, we have a program for feed efficiency called EcoFeed. We have a methane emission. We do 3D cameras for predicting body weight. And we also have robotic milking phenotypes coming. Now, with all of that, we can provide to our customers services and help them to utilize all these technologies and the genetics we produce. So chromosomal mating is a program that we have been running for a few years now that helps the farmer with their list of cows and bulls to optimize mating so they can heal the genetic progress in their herds. We have Strategy, which is a program that joins genomic and non-genomic information for the breeders so they can keep track of what's going on in their herds and see genetic progress over time. And finally, we have this last, maybe I can pick the laser pointer here. Yes. So we have the last component that I will talk about in this talk, that we are calling known-median genetic variation for now, for the lack of a better naming for it. But it's an area that is just starting within the company. We are scratching the surface, but we are already seeing a lot of interesting things, and I would like to share some of those with you guys today. All right, so moving ahead, the kind of data we have at our disposal today, we have 1.5 million genotypes available. Most of those are moderate density SNP array data, most of them with more than 60,000 markers. But we also have sequence data. One very interesting thing about our sex sorting semen technology is that for each jump of a bull, within the batch of straws we produce from that jump, we collect at least one straw per batch per bull and do a low-pass sequencing. The purpose of it is to estimate what is the purity proportion of X to Y chromosome sperm cells. So we can confidently sell a batch of semen straw saying you're buying this amount of X or Y chromosome sperm cells in that straw. The nice thing about this is over time, if we do that for the same bull several jumps, we accumulate sequence data on that bull, and then we can pull all this data together and treat it as a whole genome sequencing project. So we have today over 1,000 bulls with 5x or more. Some of our bulls have 30x, some of them 60x, and there is one or two with 80x. So that's a lot of data, and this is helping us tremendously with research. We also impute the low-pass sequencing to a panel of about 40 million SNPs. That we have some specific projects for that data, but most of the sequencing we have already performed over 77,000 sequences of straws. They are on average 0.33x. All right, so the first thing I would like to share with you guys is a program that we have been developing in the past few years called chromosomal painting, which is basically taking all the data and using the basic knowledge that we have about Mendelian genetics and chromosome theory and linkage to try to make predictions about gamete variation and progeny variation when we do matings. So I don't have to preach this to you guys because this is an audience that knows, some of you even much better than I do, all these things, but just to make the point, the breeding value, or I should say better, the direct genomic value, is the sum of the effects of genomic loci. And genomic loci are literally places or addresses on the chromosomes. We know for a diploid organism like cattle, we have chromosome pairs. One member of the pair comes from the father, the other member of the pair comes from the mother, and we call this homologous chromosomes haplotypes, so the arrangement of alleles that the animal inherited from a given parent, right? So we have the parental haplotypes, a paternal one and a maternal one. Now, when the animal makes a gamete, a sperm cell or oocyte, what they do is they take their paternal and maternal haplotypes and do crossing over and make a recombinant haplotype, and then pass that on to their progeny. So the animal not necessarily inherits the whole haplotype from the parent. It can either have an intact haplotype or a recombinant haplotype. Again, this is sounds very basic, but that's the point I want to make. So if that's all true, why should we be so limited to talk about breeding values just as a plain number, a single number? Why can't we just go back to the genome and look at those genomic effects and do something with that in terms of animal breeding? So the idea of chromosomal painting is to do that exercise. We take the phased haplotypes of an animal and we paint back to those haplotypes the effects that the animal inherited. Painting in blue, positive effects, and in red, negative effects. These can give us a picture of how the animal looks like, apart from the single number we represented, but also it gives a perspective of what kind of gametes, if recombination occurs, that the animal can make and pass on to the progeny. So we do simulations of gametes in silico, in the computer. And then we come up with what is the distribution of breeding values in the gametes that the animal can make. Now, once I have that distribution, I can just capture summary statistics from it. And if I do that for bulls and cows, I can combine the summary statistics from them and then do predictions for the distribution of the progeny. And if I have predictions for progenies from pairs of bulls and cows, I can do some kind of optimization and try to come up with an optimized mating plan. So that's the basic idea. Just to give you guys an example, this is Captain, one of the top ranking bulls nowadays in the International Evaluation for Total Performance Index. Its value is 3,200. That's the single number you will see floating around the bull summaries out there. But the idea here is take that number and see where it falls in the genome. So that's the picture from Captain's genome. Again, blue is positive, red is negative. You can see that Captain's genome is kind of a bluish, but at the same time, there are a lot of red spots in his genome. So even for a top-ranking sire across the world, we have a lot of opportunity for improvement yet. And that's the point of chromosomal painting. Just to give you a contrast of an animal that has a more of a reddish genome. So this guy, we even censored its name for its privacy. We have a lot of reds everywhere, but still an animal with a bad breeding value like this, it can also have blue spots in its genome. So it's just to show you guys that this is not only plotting noise and things that will not be replicable. This is clearly showing the contrast between two animals that have divergent breeding values for a given trait. Now, just showing you another trait. So this is milk yield. I plotted two animals here. These are their chromosomal paintings. And the idea is that once I get this, I can recombine using, of course, a recombination map telling me where in the genome I have recombination happening more frequently. So I can do a good job in simulating those gametes. If I do a few thousand of those, I can plot a distribution. So that's what we see here in panel B. This is the distribution for chromosome 1, for example. This animal happens to make pretty average recombinants for chromosome 1, but here's where things start to get interesting. If we jump to chromosome 14, for example, you can see that mostly this animal will show very bad chromosomes 14s to its progeny. But in rare occasions, it can also make very exceptional breeding values for chromosome 14. Why is that the case? Because we have QTLs in the genome. So chromosome 14 is a very famous case. That's why I'm bringing that up to you. You probably know about DGAT1 and its strong effect on the number of traits that are relevant to dairy cattle. But this animal is heterozygous for the variant at DGAT1. And if it gets the rare chance of aligning that strong effect with a lot of blue from the other haplotype, you get exceptional chromosome 14s for an animal that otherwise you would think could only shoot bad chromosome 14s. The same way we can look into this other animal, it can make very good chromosome 5s, for example, because it has a very strong blue effects for some QTLs here and there. But it's also heterozygous for chromosome 14. So even though overall it makes very good chromosome 14 recombinants, sometimes it can make pretty bad chromosome 14 recombinants. The consequence of all of that is if I just take all those distributions and collapse them into a whole genome distribution, we get things like this. So for this first animal, we have a right skewed distribution. And for the bottom animal, we have a left skewed distribution. If you look at excess kurtosis, which is measuring the weight of the tails, you see that it's a negative excess kurtosis, meaning it has a heavier tail than a normal distribution would have. If you know anything about a normal distribution, you know that it's also symmetric, so it shouldn't be skewed. And we clearly see that gametes do not need to have normal distributions for some traits, especially if there are major QTLs influencing that trait. That's a very important message because we have the tendency to, as a community, sometimes to get about our day, to do some assumptions in our statistical models that may not hold in practice. Even for a model like the one I'm presenting to you, this is all based on effects from a SNP kind of model. And we know that this type of model does a very heavy shrinkage on the effects. It's not letting QTL effects stand out much. Even in that situation, we still have a heavy influence from the QTLs here. So I think that's a very important message from this program. Now, the next step into that exercise is I do that for a bull and a cow. Can I predict progeny? So this is a case of two animals, a cow and a bull. This is the distribution of oocytes for this cow for milk yield, for example, and the distribution of sperm cells for this bull. You already can see differences between individuals. So the oocytes of this cow, they have a large variation, heavy tails, and a slight skewness to the right, whereas the bull looks kind of a normal distribution with skewness and kurtosis, excess kurtosis, very close to zero. Now, if I combine those two guys, this is the prediction of the distribution we'll get. And the nice thing about this particular bull and cow is that we produced 42 real offspring for them. to do a validation. So these offsprings, these are the genomic PTAs we produce for those offsprings, and they are plotted here as a rug plot under the histogram. Males are in blue, females are in pink. You can see that they all fall within the range of the distributions we predicted. We made a slightly larger validation for these three traits as well, looking into 122 families, comprising 44 bulls and 103 cows. Each of those pairs with at least 10 progenies each, in a total of 1,500 progenies. And you guys can see here what we predicted and what was realized. And the histograms, they align pretty well, and correlation is very high. So we're very happy that we can do some nice predictions about these matings. The implication of that is for a program like the one we have for GBIT, for example, if I'm pairing a heifer with a bull and I have the distribution, I can estimate quantiles from that distribution and know what are my chances to get a PTA at a given threshold. And even if I get a probability like 10% of getting that threshold, I know that out of 100 embryos on average, 10 will meet that criteria. So this is the kind of a lottery game we can start playing if we know all this information. All right, so all that is exciting if what we know from basics holds, but we know that biology is a crazy field and things can get off script very easily. So I'm going to show you some cases where that assumption of paternal and maternal haplotype can start breaking. So the first case we have is what we call uniparental disomy. If you're analyzing SNP data and you see a big chunk of a chromosome that is all homozygous, the first thing you would think of is autozygosity or inbreeding, meaning that the paternal and the maternal haplotypes within that region, they are identical by descent. Now, what if I delete a piece of a chromosome or an entire chromosome? The SNP array doesn't know that you're locking a chromosome. It's just doing probe hybridization, and it doesn't know locations in the genome. So it's just giving you genotypes. If you look at a deleted chromosome, it will look like a run of homozygosity as well and could be confused with inbreeding. But we could differentiate these two situations by doing some extra analysis. So the first one we could do is to compare the genotypes of the offspring with the parents. In the case of missing a whole chromosome, you would see, for example, let's say that the paternal chromosome is missing. If I compare the genotypes of the offspring with the paternal genotypes, I would see a lot of Mendelian mismatches. So alleles that the parent has or the animal has that couldn't be passed through Mendelian segregation, right? So that's one thing that we can look into. But if you're really missing one copy of a chromosome, the nice thing about SNP array data is that it behaves as a array CGH analysis as well. And you can see or do inferences of number of copies of chromosomes. So you would see a lower probe intensity data for that single copy chromosome as compared to the 2 copies for the rest of the genome. The uniparental disomy is a different condition where instead of having each haplotype coming from a different parent, I have 2 haplotypes coming from a single parent. So either 2 paternal or 2 maternal haplotypes for a given chromosome pair. Now, how to distinguish this from the other 2 cases? So we would still see runs of homozygosity, and we would still have Mendelian errors with the parents that did not contribute with the haplotype, but the intensity data would be normal, right? And these phenomena, they are reported in the literature in an opportunistic way, like there is an instance of a case where a person or an animal had a genetic problem or a genetic disease. They investigated, they found out that it was the subject was missing a piece of a chromosome or had two haplotypes from the same parent or things like this. But what about animals that have no syndrome or have no associated phenotype to them and they look perfectly healthy? Can they also have something like a uniparental disomy or at least a partial uniparental disomy in their genomes that would break our assumptions that we had before about haplotypes coming from different parents? So this is a case where that happened. Male calf that is happily healthy, living and grazing and all that, that had this condition. So if we look at the heterozygosity across its genome, basically when we see a dive here around 0, it means a runoff of homozygosity in the chromosome. We can see that on chromosome 2, 10, 15, and chromosome X as well, because it's male calf. The chromosome X, the calf has just one copy. The sire contributed with a Y chromosome, and the X is coming from the mother, right? The only strange thing we see on chromosome X is this part over here, which is a special region on chromosome X called the pseudo-autosomal region, which has homology with the Y chromosome. And in this SNP array, it can look like it had heterozygosity on the X chromosome. But that's perfectly normal. That's what we expected to see for chromosome X. And the nice thing about the calf, in this case, a male animal, is that we can use X as a control for our analysis within the single sample we are analyzing. When we look at the Mendelian mismatches with the sire, for example, we see across all the chromosomes very low Mendelian errors. The little bumps you see here and there are just random genotyping error. When you look into chromosome X, you'll see this huge wave of points going up, meaning that the X chromosome came from the cow, not from the sire. But what catches our eyes is chromosome 15. And the peak we get is matching with the runoff homozygosity we just saw in the last plot. Now, we have to differentiate between, has this animal deleted part of its chromosome 15, or do we have a uniparental disomy? When we look into the probe intensity data, everything looks normal except X, which is single copy. It's a little below from the rest of the genome, but chromosome 15 is just fine. So the animal has two haplotypes for chromosome 15, but one of the haplotypes has a piece that is not coming from the sire. The nice thing about chromosomal painting is that we can also see this kind of pattern happening. So this is the calf. This is the maternal haplotype. We can even map back to the dam haplotypes and see what kind of recombinant haplotype was made for that calf. The beginning of the other haplotype matches with the sire, but there is a whole 2/3, let's say, of the chromosome that is not coming from the sire, and the sire has nothing to do with this kind of pattern in chromosomal painting. So whatever happened in that calf, it just copied the other haplotype it received from its mother. So this is a case of uniparental disomy. Now, another interesting exception that can happen is what we call a balanced translocation. So just going, yeah. Let's think about, just to simplify matters here, two chromosome pairs, chromosome A and chromosome B. Translocation is an event where a chromosome breaks, and during repair, it gets stapled together with the wrong chromosome, right? So for example, I took chromosome A, I broke it, and I broke also chromosome B, but instead of putting them back as B and A, I made two new derived chromosomes. I'm calling them derived 1 and 2, which they're kind of a recombinants inside quotes, but we call this a translocation, right? So the cell will try to make homology out of this thing and do the pairing and make sure that mitosis is going fine. So we may get this kind of aberrant karyotype in this case. So chromosome A, for example, pairing with derived 2, and chromosome B pairing with derived 1. The problem of this is if we are doing karyotyping in the lab, all we do is to take images from condensed chromosomes and putting them together in order by size. And we may put the chromosome A with derived 1 and think this is normal chromosome A. And the same with chromosome B. I just put them together because they are roughly the same size, and I falsely infer that this is a normal karyotype. So that's the kind of balanced translocation that I'm going to show you guys. Let's think about this animal with this kind of translocation. So apart from the differences in masses that are happening with the new derived chromosomes, we also have chromosome X and Y in the case of a male. What kind of gametes I could produce out of that genomic configuration? So I could have things like the two normal original chromosomes with X, the normal ones with Y, now the derived with the normal and X, and so on. And if I just put them all in order from the lightest gamete I can produce to the heaviest gamete I can produce, I have this gradient of DNA mass for the sperm cells they can produce. Now, again, ST is a company that has in its core a sex sorting technology based on amount of DNA and mass of sperm cells. So it means that our technology can see this before a karyotype or a sequencing project tells us there is something strange with a given animal. So this is an example of the kind of output we would see in the program we run in our machines called CaryoFlow. But this is the fluorescence in 2 dimensions. This is the population of Y chromosome cells and the X chromosome cells. We don't need the details or a specialist to just tell you that we can see 2 populations of things and we can separate them using the flow cytometry. If we just turn this into a histogram or a density plot, it's better to say, I would say this bimodal kind of distribution with the Y and the X chromosome. But if I have a translocation, I would start seeing different kinds of patterns in the histogram and the scatter plot. So if the mass differential between the derived chromosomes is small, I get this kind of a fuzzy and merged distribution where the valley here in the between the 2 peaks gets a little bit shallow or superficial. Right? Whereas if I have a huge mass differentiation between the derived chromosomes, I can even get multiple peaks in CaryoFlow. So what we can do to validate that is, since we can select the subpopulations from whatever place in the plot we are using our proprietary technology, we can go to the extremes of the distribution, collect those sperm cells, and do sequencing on them. I must say that it's very difficult to have a pure mixture of just the derived chromosomes. You will get some of the normal cells as well. So we get a percentage of purity that is very high, but there is some still left. So it's going to be a little mixture of things, but still we can clearly see the difference between the 2 populations. This is a case of an animal that we ran CaryoFlow for some semen straws on it, and it had this shallower pattern in the peaks and a mixture of the populations in the scatter plot. So this is telling me that there is a mass differentiation between the populations and most likely balanced translocation for that. Now, the nice thing about carrier flow is that based on where those populations are in the plot, I can estimate the percentage difference between the two populations. In that case, carrier flow told us there is a 7% difference in mass between these two populations. We went on to do the karyotype of that animal, and we got a normal karyotype. Again, because this is just sorted by size, and we're not seeing the translocations because they are mimicking the original sizes of the source chromosomes. Now, we did also whole genome sequencing on blood samples from that animal and ran pipelines for structural variants identification. And we ended up with large lists of false positive things. It was really difficult to just set up a pipeline and say, I'm going to screen every single sequence I receive for balanced translocations. It won't happen. From our own experience, it wasn't easy. If you don't have an indication of where to look at, it becomes very, very difficult. Because again, it's a balanced translocation. You don't have any difference in genome content. And the reads you produce, they are based on inserts, so fragmented DNA. Unless you do long read sequencing, we also did low pass long read sequencing. There you could see better, but still it's kind of hard to navigate through all the data. So by taking the populations with differential mass, just contrasting the depth of coverage we got from the sequencing, we can see that this is a contrast between the upper tail, so the heavy cells, against the lower tail, the lightest cells we got. So the heavier ones, they would have the X also, and the lighter ones would have the Y chromosome. But apart from that, you can clearly see a signal coming from chromosome 2 and chromosome 14. And when you do the upper tail minus the lower tail, you get more chromosome 2 and less 14, and conversely, less 2 and more 14 in the other way. So if we just screen through that data using basic models like a basic hidden Markov model just to pinpoint the place where that translocation happened, we get a very precise position that we can go back to the alignment data and now see what's going on. So this is a screenshot from IGV. If you're not familiar with that, just walk you very quickly through it. This is the coverage of the region plotted over here. Each horizontal bar here is a read in gray, and the reads that are in orange here, they have their pairs mapping to chromosome 14. If I go to the location where they're mapping on chromosome 14, I see some red reads, which are complementary. Their pairs are mapping back to chromosome 2. So it's a crosstalk between these two positions. And these colored places in the reads that you guys are seeing here is what we call the soft clipping. Soft clipping is just a mismatch at the end or beginning of a read between the sequence that you have in the read and the sequence that you have in the reference genome. If I take these bases that are not matching with the reference over here and blast them against the reference itself, they will map to that position in chromosome 14. And if I do the same with the soft clipping here, it will map back to there. So we have a very clear cut where the breakpoint of the translocation is. And with that, we can estimate the sizes of the derived chromosomes. It was very nice to do that exercise because when we compare in terms of percentage, the percentage difference we got from sequence data is 7%. Again, confirming what carrier flow is telling us. So carrier flow is a very powerful tool, and we have been using it to screen for this kind of events. And for our surprise, they are not that rare. We already have accumulated a number of cases of bulls that have this kind of a translocation. I think confirmed with karyotype and in situ hybridization, we have about 6 cases. This animal over here, the only associated phenotype we have for it is a lower conception rate compared to contemporary bulls. So its conception rate is about 29% against 45 to 59 for its contemporary bulls. But apart from that, this animal was a high-ranking genetics bull that we could just use and not knowing that this was going on. And the most surprising part of it is that this is heritable. So there are animals that can inherit those translocations, and because they have a balanced genome content, they would just go about their days just fine. So this is the case where things that we know by heart may go off a little bit, even though it's a small frequency in the population, it happens. And finally, the other thing that I wanted to share with you in this, I'm calling non-Mendelian genetics, but just for the sake of saying that things are not the way we expect them to be, I would like to show you a case where gene content diverges from what we expected based on a reference genome. So this is a bull that has a color dilution phenotype, and this gene, the name gives away some information already. So it's a melanocyte inducing transcription factor, MITF. It's one of the essential genes in the cold color pathway. It's widely known, very studied in many, many different species. But what is very interesting about this gene in this bull is that if you look into the alignment data for that bull, in that gene, you see this paired end reads. We are connecting now the pairs with a straight line. You see that the gap between the two reads is exactly the coordinates of the introns of the gene. And here I'm plotting different isoforms from the same gene. And MITF is a very complex gene with different isoforms that are expressed in different tissues. It has different promoters for those different tissues. So sometimes we as a community can be quite naive in saying, I'm going to explore functional variants because they are changing an amino acid and these kind of things. But genomics is much more complex than that. And when we say function, it can embrace a number of things that goes way beyond just protein coding genes and changing amino acids here and there, right? So this is a clear case of that. We are far from having annotated all the isoforms for that particular gene, but we have a good representation of what this gene can do in terms of variation in expression. But going back to the alignment, we see that the reads are aligning perfectly with the exons, and between exons, we have nothing. Of course, I have more reads on that animal. I'm just showing you guys the ones that are jumping exons, so we can highlight those. But what is going on here? So the sequence is telling me that I have at least one copy of this gene, the genome of this animal, that has collapsed the exons together and skipped the introns. So we know that messenger RNAs, when they are produced and they're processed into a mature messenger RNA, they go through a process called splicing. So they lose their introns and their exons are put together. Now, there are elements in the genome that we call mobile elements that they can encode for reverse transcriptases, which they can take RNA and transcribe back into DNA. and that DNA can paste itself in another place in the genome. So this is what's happening with this animal. It made an MITF transcript, it got reverse transcribed and pasted back elsewhere in the genome. If we just zoom in into the exons of that animal, we can see a very interesting pattern. So these are the red reads, some normal reads here and there, saying that that animal has the normal 2 copies of MITF, but now with an extra copy somewhere else. We can see some evidence of cleavage increase around the exons as well. And if we take the soft clipped reads and blast them from exon 1, they will map to exon 2. If we take the soft clipped reads from exon 2, map back, they will go to 3, and so forth, so on. This is telling us again, this is just a messenger RNA that was put back into the genome. If we zoom in into the beginning of the gene, we see colored reads. It's a dark gray or purple, if you will, read. If we click on it in IGV, it will tell us where its pair goes, and MITF is on chromosome 22, but the pairs of these reads are going to chromosome 12, where the retrocopy pasted itself. Now you can say, well, that's one case. Maybe this is not something normal and may not happen very often. But here's the sequencing I've done in dog. I have other projects outside of ST, research projects going, still going, that I started before joining ST and haven't been able to deliver the paper yet. But one of the projects that was involved, we sequenced a few animals for cold color studies. And we know in dogs that there is a retrocopy of MITF on chromosome Y in some breeds. And one of the dogs we sequenced is a carrier. So that's a screenshot from that dog. What you see is the exact pattern we just saw for that bull. So it's not something abnormal or something it's not happening. So MITF seems to have a history of liking to do retrocopies of itself. What this does to our gene annotations and gene content assumptions, well, that can make things a little bit more complex than we would like them to be in our analysis. All right, so with that, concluding the presentation. So we have chromosomal painting as a new tool that we are working on, and it seems to be working nicely, but it has some assumptions from the basics we know, right? And some of those may not hold in some cases. But the nice thing is that the technology gives us the opportunity to see some of those exceptions, like the uniparental trisomy, um, balanced translocations, and even differences in gene copy. A nice message about this is that there is no single analysis or single software or automated pipeline that can just give us all this information. You have to do a lot of different analysis. You have to look into the data very carefully, open up sometimes regions in the genome, genotypes, alignments, and not just rely on whatever is the VCF file or whatever you got from the automated pipeline. There's a lot of manual work that still needs to be done in order to understand better the data. But of course, there are some screening efforts that we can put together, at least to highlight possible cases within the data sets we have. So with that, I can give the word back to you. And if you guys have questions or things you want to discuss, I'm more than happy to. I'm back.
Thank you. We'll now open the floor for questions. If anyone has any questions, feel free to raise your hands, turn on your microphones, or if you'd prefer, you can write your question into the chat as well, and I could read it out. Larissa, go ahead. afternoon. Can you hear me? Can't hear you, Larissa.
Me neither.
Okay, let's go. While Larissa writes, let's go to Jose.
Hello, good afternoon,
Hello.
everybody. First of all, thanks for the invitation. Easy sent me a last-minute invitation. It was a pleasure to listen from Yuri's work at Sexing, and it's fantastic to see how we find new features in the genome that mess up all our thinking. So when we just estimate the power of a SNP, realize that we can have this investigation. So I would like to just congratulate Yuri for this presentation and send my regards to Flavio that should be also here. I saw his name and say that a long time I miss you from Aracatuba. So just a comment. Thank you.
Thank you.
Thanks, Fernando. So for those who don't know, Fernando was my supervisor for my master's, and my PhD is the person that
No, I
started
was
it
there
all for me.
just giving some support. He did everything by himself. He is the guy.
So I own much of the things I know today and have done in the past to this guy. So if you guys don't know, he was the person that helped me the most in my career. So thanks, Fernando.
Okay, I was wondering. Oh, I think Larissa is here. So Larissa asks in the chat about the SNP probe density. Is that log R ratio, or is it another measurement?
Right. So it was supposed to be log R ratio. The thing is that in order to calculate log R ratio, you have to have your clusters. If you guys remember the plot, when you do like the contrast between A and B fluorescence versus the total intensity, you have to have a training set where you look into a centroid and an ellipsoid. You train a model and you know the AA and B and BB clusters. And from there, you have expectations of what would be the contrast and the intensity values for the different clouds of points for the three different genotypes. With that, you have an expected R value and then observed R value. Then you can take the log 2 R ratio and do the calculations. For that animal, we had a genotyping, the Thermo Fisher platform that we could still calculate log R ratio, but we would need to analyze some training samples together. And we were kind of in a rush to look into that animal because it sounded as an interesting project to do. So I just did the log R, not the R ratio. You can think of this as something very similar to what we used to do with our ACGH before genotyping came along. People would do just the log R, not the log R ratio. But they are kind of similar metrics. The log R ratio, of course, has much less noise. But even at that noisy level we saw in the plots, it was still pretty clear that only the X chromosome was a single copy one.
Okay. And just maybe a more broad question I was wondering, since you seem to be up to date on many different technologies, are there any kind of latest like trends in research or specific areas in research that are kind of promising next targets that you kind of are interested in translating into application?
Oh, yes. There are things that I can't really talk about because this, things that we've been doing. But I'm very excited about many different things like 3D chromatin confirmation. This has a role already known role for some model species and humans in the function of variants and how the expression works. We can have, for example, QTL influencing the expression of a gene that is very far away from it, just because when the expression is happening, DNA is put into a loop like a shoelace. And those regions that look like very distant in the reference genome, they actually physically touch when expression happens. This is an area that's very exciting. I'm just talking about within genomics, right?
Yeah.
So methylation and genetics in general, these are topics that are very promising, and we should expect nice technologies coming from those areas in the near future.
Yeah, very interesting. Thank you. So it looks like we're out of time. Thank you again for the very nice presentation. And thanks everyone that joined us this week. We'll see you all not next week, but the following week we'll be back. So thank you.
Thank you.
Bye.
Bye-bye.